While dendrites can generate graded potentials, somas can generate graded potentials, axons only propagate action potentials. That's it. Okay? If there is not enough of a potential difference across the membrane of a neuron to reach AP, the axon doesn't do anything. Okay? But if there is graded potential to threshold, the axon enters AP. This AP does not lose strength, even if the axon is 3 feet long. Based on the way ax potentials work, it never loses strength. Once generated in the central nervous system, it will continue with the same strength, the same magnitude of depolarization and repolarization. Doesn't matter, anywhere along its length. So the strength of an ax potential always remains the same. Okay? There is no endoplasmic reticulum, Golgi apparatus in the axon, and so the axon has to rely upon the nerve cell body to provide substances that it needs in order to maintain its physiology, communicate, do ax potentials, resting membrane potential, all of that. So no surprise, if you remember this right here, when you get into your clinical practice and somebody asks you about, well, his nerve's cut, will he ever be able to get it back? Remember that those axons now have been deprived of their source of materials because the axon has been cut and severed away from the nerve cell body. It can't get what it needs to maintain function. It malfunctions. Okay? All right. Got about 10 more minutes left. This is helping you understand then the fact that axons, whether in the central nervous system, peripheral nervous system, oftentimes then are modified with this thing called the myelin sheath. Segmented sheath around most long or large diameter axons are referred to as myelinated fibers, okay? And there's actually a color difference between myelinated fibers and unmyelinated fibers, or what we call non-myelinated fibers. And this is something that is actually visually something you can see in dissection. You can see when there's a large dominance of myelin in a nervous tissue and when there isn't, okay? Myelinated tissue looks whitish, yellowish due to the presence of fat there, okay? So what you're seeing here, no great explanation needed. You're simply seeing that as an axon is being built, Schwann cells in the peripheral system will surround the axon. I would point out this area right here, and that is the cytoplasm of the cell is actually eliminated from the overlap point in this area. The cytoplasm instead resides in the most peripheral portion of the Schwann cell. Schwann cell is a living cell, okay? But there is no need for cytoplasm in there. The primary purpose of this wrapping is for insulation protection, okay? All right? What does it do? Protects and electrically insulates the axon, and this is what we'll focus on, increases the speed of impulse transmission, okay? When we talk about the need to make adjustments rapidly, more than likely, the effector is going to be serviced by a myelinated axon, okay? So if I'm walking down the road and I trip over something and I raise out my hands and I change my body position, considering that it's occurring in a split second, okay, pretty much, so I don't fall on my face, we're talking about myelinated axons that are used to get the information to and from the central nervous system. And then even in the CNS, myelination is used, okay? So that's the big thing there, is it speeds up speeds up the rate of impulse transmission. This is an electron micrograph. You can see 24,000 times magnification. So there is your axon's diameter, but it's 24,000 times magnified, so we're talking about something that's really small. And then you can see how the myelin really does consist of simply the phospholipid bilayer of the Schwann cell. The rest of the Schwann cell we can see is out here, okay? But that is phospholipid bilayer. Not much cytoplasm there. The primary job is just simply surround, okay? This is something that comes from your book. And now that we've considered body, dendrites, axons, we look at different kinds of neurons. Your time spent on this diagram will be well spent. The first thing we see is that there are structural classifications, okay? We are then going to take the neuron and organize it into 3 regions. And this is important and worth your time as well, okay? So if we start at the top, structural class, multipolar, bipolar, unipolar, or pseudo-unipolar, okay? Let's look at the major difference. By the way, bipolar has nothing to do with the disorder. It has to do with the structure of the neuron, okay? This is a multipolar neuron. Many processes extend from the cell body. All are dendrites except for a single axon. So there's your definition of a multipolar neuron. I still don't know the derivation why we name it that. Bipolar. 2 processes extend from cell body. One is a fused dendrite, the other is an axon. So in this situation, they're calling that a dendrite, and then it has kind of a bunch of, I wouldn't say necessarily endings, let's call it, sometimes these are referred to as free nerve endings, but they're more like free nerve beginnings, okay? Because that's where the impulse comes from. You'll notice then that there isn't a lot of branching off the body. It's one basic branch for dendrite, one for axon, and then off it goes. So bipolar seems to work. There are 2 poles on this cell. And then pseudo-unipolar, okay, or unipolar, again, we have these areas here known as receptive endings, but this whole thing here is referred to as an axon, okay? And the nerve cell body then is somewhere off to the side. When we looked at satellite cells, what we were seeing were nerve cell bodies of this kind of neuron being surrounded and protected outside of the CNS. Okay? No big deal here, all right? Just different kinds. This one's rare. This is rare. Found in 2 places that I know of. The olfactory epithelium, sense of smell, and in the retina of the eye. I don't know of any other places. There probably are a few others, but they're rare. So not a real common type. This one, very common. This one, most common. The multipolar neuron is the most common neuron in the human system. Now let's try to figure out what the heck we're seeing here. The relationship of the anatomy to 3 functional regions. The regions are given as receptive region, as a stimulus accepting area. The conducting region, a region of the cell that conducts an impulse. And the secretory region, where neurotransmitter is released to pass that impulse to the next cell. As we can see here on a multipolar neuron, the nerve cell body and all of its dendrites are considered receptive. This is where other neurons can send their impulses and communicate with this multipolar neuron. In the green area here, we're talking the conducting region. This is where action potentials occur. Graded potentials occur in the receptive area. This is where multiple neurons can release their neurotransmitters and affect the polarity across the membrane of the cell, maybe trying to push it towards AP or maybe trying to inhibit it and push it away. But if AP occurs, it's going to then travel down this trigger zone, however long that trigger zone, however long that conducting region is. That's where it's going to travel until it finally reaches the axon terminals where it secretes its neurotransmitter. If you take a look at the bipolar neuron, it's not that much different. It's dendrites and soma that are the receptive area, and then the axon is the conducting area, and then the secreting area then axon terminals. Then we'll end with this because you guys are ready to be done. This is the one that's sort of interesting, and that one is the receptive area is an area of free nerve endings. This is the type of neuron that is going to provide pain sensations in your skin. because this receptive region of this neuron is right up under the epidermis. And if you bang it, smash it, cut it, poke it, you're going to disturb these things, and an action potential is going to be sent to the CNS. You'll notice that this area is called the conducting zone then, and this was our axon. Axons are typically where we see conducting zones. Okay? So remember table 11, it helps us to understand the difference between, as well as then, how the cell receives info and sends it along. I'll see you in the lab here in about 15 minutes or so.